Good morning, everybody. Welcome to the webinar. We're just going to let everybody trickle in here and we'll start a minute after 8. Good morning, everybody. We're going to start the webinar in a minute or so while everybody trickles in. Okay, good morning, everyone. We're going to get started by going through some introductory slides before we move on to our main speakers today. So welcome to the RCN webinar for November. This is a monthly webinar series. Today we are having a special Swiss episode of the RCN. We're going to hear from Adrian Lison and David Dreifuss in a moment. And first, I have a few announcements to make. So first, for those of you who are interested in our students, please, and have students, please pass on this QR code for Discord, which is a sort of like Slack. And Tamina has put information in the Slack, in the chat about it. We have 3 ongoing activities. We have a monthly journal club. We have the webinar, and we have nominations available for the webinar and an email list. So if you would like links to any of those, Tamina is putting those in the chat. And we are starting an RCN participation certification program. If you have questions about this, please email Tamina, and you can scan this QR code, and we'll also put a link in the chat as well. And then finally, just a plug for our December series, we'll have 2 speakers. Liang Xiao from Michigan State will be talking about sexually transmitted infections in Detroit wastewater. And we will also have 2 speakers who published on the influenza sequencing in wastewater in New England Journal of Medicine. They will be speaking as well. So that will be a good chance for those of you interested in influenza sequencing to ask all your questions of that speaker. So with no further ado, I'd like to introduce our first speaker, who is Adrian Lison, who is a PhD candidate at ETH Zurich in Basel, ironically, not in Zurich. Adrian, you can go ahead and share your screen while I just remind everybody that we are moderating questions in this webinar. So when you have questions, please put them in the Q&A, and then I will be moderating the questions for Adrian. Thank you, Adrian, for speaking today. Go ahead.
Yeah, thank you very much for the opportunity to speak here. So as you already said, Ali, so since we're having a bit of a Swiss session today, I would first like to start by giving you a short background on the project that both me and also the second speaker, my colleague David, are involved in, which is called WISE, or Wastewater-Based Infectious Disease Surveillance, and is led by this great team of PIs. So on the computational side, Tanja Stadler and Nico Bernwinkel from ETH Zurich, who also are mine and David's supervisors respectively. And then Tim Julian and Christoph Ort on the environmental microbiology and engineering side at ERWAG, which is the Swiss Federal Institute of Aquatic Science. And then also Tamar Cohn from EPFL in Lausanne on the environmental chemistry side. And so this consortium, like many other initiatives, really started during the COVID-19 pandemic with monitoring of SARS-CoV-2 in wastewater, but has now evolved into a research project on methods both on the computational and on the laboratory side for wastewater-based epidemiology. And it's also running the current wastewater surveillance in Switzerland, which has since been extended to also monitor influenza A and B virus in the wastewater and RSV. And we're currently doing this for 14 different wastewater treatment plants across Switzerland using 24-hour composite samples from the influent wastewater, which are then quantified for the respective targets using a multiplex digital PCR assay. And in addition to this, some of the samples are also sequenced for genomic surveillance of SARS-CoV-2, which my colleague David will tell you much more about later. But now for the general monitoring of transmission dynamics of these different pathogens in the wastewater, we are computing the effective reproduction number from the concentration measurements on a weekly basis. And for this, we're using a model that I've been working on for quite a while now, which also exists as an R package called EPISUR. And the main idea of this model is that we describe the full so-called data generating process. So starting with the transmission process and the new infections, all the way to the actual data that we are observing, which are concentration measurements in one single statistical modeling. So we basically have a number of different mechanistic components in this model that describe how the data that we're actually observing is generated. And so this single model can be used to simulate data, but we can also use it for Bayesian inference by fitting this to observed measurements and then estimating a posterior distribution for like our parameters of interest, like the reproduction number. So really the idea is that we have just one whole model that describes all the aspects that we think are relevant in generating these wastewater data and that we can then use for inference and directly fit to concentration measurements without any additional pre-processing steps. So now what I would like to talk about is not really any specific details of this model or the package, but I would rather want to point out 4 main reasons why I think this approach of generative modeling is quite suitable for wastewater surveillance. And I will try to illustrate this with our experiences from our modeling, but I think it applies more generally to wastewater-based epidemiology. So the first argument is related to the fact that wastewater measurements are inherently noisy, and we typically need some kind of smoothing to actually identify a relevant trend in our time series data. And in the beginning of our monitoring of SARS-CoV-2, we started using with a different method developed by a former colleague of ours, Jana Hussmann, which was it's a stepwise approach And in this approach, we basically smooth the time series of wastewater measurements using local polynomial regression, the lowest method, which is like a very well-known method used for smoothing time series. And this method has one important hyperparameter, which is called the window length or window size, and basically describes the number of days included in the smoothing window. And this really determines the strength of the smoothing. So now when we're applying this method to our wastewater data in the beginning, a difficulty is to choose this parameter because we don't really have a reference. We don't really know how smooth we really expect the time series of measurements to be. And so we can't really do some kind of hyperparameter optimization. So basically, we ended up making a rather arbitrary choice of 3 weeks. And this seemed to work well with the SARS-CoV-2 data that we had at the time. However, after a while, we basically observed that our reproduction number estimates had become quite volatile. And we were suspecting that this was because our sampling frequency was reduced at some point from 7 days per week to 5 days per week. So we tried out longer smoothing windows, and you can see that here this makes quite a relevant difference for the reproduction number estimates that you get. And fortunately, at this point in time, we also had the generative model that I just showed you before in place. And by comparing the estimates from this model, we then figured that probably a longer smoothing window was now more appropriate. But as you can see, it's quite difficult to motivate a choice for the smoothing parameter because the window length doesn't really have a clear epidemiological interpretation. And we also found it can change over time. Now, in this generative model that I showed you before, there's also non-parametric smoothing, but it's at the level of the reproduction number. So we're using some time series model, for example, a random walk model or exponential smoothing model or like smoothing splines to describe how quickly the reproduction number can change from one day to the next. But the advantage here is that on the one hand, it's a bit more interpretable because we can really think about how volatile we expect transmission dynamics to be. And then the other point, more importantly, is that this is really independent from all the other factors in the data generating process. So for example, it's independent of the shedding load distribution or of the actual measurement noise or the sampling frequency, which are all factors that influence the apparent smoothness of your data, but they're not relevant for the smoothness of the underlying transmission dynamics. And we find that this actually makes the smoothing that you use less critical in the end. So here, for example, you can see a comparison of estimates from this generative model when we either use a random walk smoothing or when we use spline smoothing, which are quite different smoothing approaches, but they differ in terms of uncertainty that we get, but generally have a very similar shape. The next point I want to highlight is very closely related to this, and it's basically that we, in wastewater surveillance, we often just have a couple of samples per week. So we have dates where we don't have measurements and we kind of need to interpolate data there. And the nice thing about these generative models is that we basically get our interpolation for free because we can basically just leave out unobserved dates from our likelihood and then just apply the approach like we did before without any additional pre-processing or interpolation. And ideally, this also accounts for the increased uncertainty from the lack of data. So here you can see, for example, in the comparison we did where we had in gray, like daily samples, and then in the different colors, really just one sample per week. And you can see that this really loses some detailed signal on the transmission dynamics, but the overall shape can still be retrieved. And when comparing this again to this other method that we've been using, it seems that using this kind of generative approach is a bit more stable because in the other method we had really needed some interpolation step, and this could increase the dependence on what weekday we are sampling, and also in the generative method, really the increased uncertainty seems to be better represented. The third point I want to highlight is that in generally environmental surveillance, we are measuring concentrations using a well-known quantification method in a laboratory setting. And this really provides some potential for also mechanistically modeling the actual measurement process. So we try to do this a little bit for digital PCR, which, as many of you know, is based on partitioning your analyte in a large number of different chambers or droplets, which each have an independent PCR reaction. And then from the percentage of positive partitions, an absolute concentration can be computed. And the nice thing about digital PCR is that it has some really clear defined partitioning and count statistics that you can use to predict the statistical properties of your measurements. And so what we did, for example, we compared the theoretical predictions for the so-called coefficient of variation of the measurements, so describing basically the variation relative to the mean, and the probability of getting a non-detect, like a zero measurement, and compared this to empirical estimates that we just get from our surveillance data. And one important thing that you can see here is that if your basically all the concentrations in your data are really high, then you're probably in a regime like this, where basically the coefficient of variation is approximately constant. And in this case, it's really justified to, for example, apply a log transformation to your data and then assume that it's normal distributed with constant variance. And this is a really widespread approach to then, for example, apply ordinary linear regression on the data and so on. However, if you also have lower concentrations in your data, then this coefficient of variation is actually rather concentration dependent, which means that even after a log transform, you will have heterogeneous variance that depends on the concentration. And this can invalidate some of the assumptions in a regression, for example. Also, at these very low concentrations, you have a high probability for getting those non-detects, the zero measurements. And here, a problem is that if you like dropping those non-detects from the data or just replacing them with some value, like the limit of detection, can also lead to some bias. And in this preprint that we recently put out, we also looked at how these dependencies depend on certain laboratory parameters, and also on how much variation you have in your lab process before the PCR. So for example, due to noise in the extraction process, this can really influence like the actual relationship in dependence to the concentration. So now with respect to the generative modeling, what we try to do is then we basically developed a mechanistic model that accounts for these statistical properties and allows us to directly fit to the concentration measurements without needing to account for a limit of detection or a limit of quantification, because this is already covered in this statistical model. And then when comparing this to simpler models, we find that this can like improve your model fit, especially if you have both low and high concentrations in your time series. So finally, the last thing I would like to point out, which I think is also a strength of generative modeling, is that it allows some kind of out-of-sample validation. And so what I mean by that is, if we go back again to this model that I've shown, when we're estimating the reproduction number, we ideally would like to be able to evaluate how good our estimates are. The problem here is that we don't really have a ground truth. We don't know what the true reproduction number was. And so this is really a limitation. However, something that we can do is, I said before that we use, for example, a random walk model to describe how the reproduction number evolves over time. And we can actually extend this into the future to basically make a kind of forecast of how the reproduction number will evolve, which by default assumes that it will stay at the current level in expectation, but there's also increasing uncertainty into the future. And you can also do this with other approaches, even also with penalized splines, although you have to be a bit more careful there. But the general idea is that we can produce a forecast of the reproduction number. And once we have that, we can apply this whole generative model to simulate or basically predict future concentration measurements. And now these are really things that we will observe in the future. So what we can then do is we can retrospectively assess how well our model was able to predict short term those concentration measurements. And we can use various forecast metrics and scoring rules for that to get an idea of how well the model was able to basically estimate the current growth trend in infections. And this is, of course, not directly the same as evaluation of the, say, reproduction number estimates, but it's maybe the next best thing we can do to get an idea of how meaningful our real-time estimates from such a model really are. And this is possible or really has a meaning because in this generative model, there's always a direct correspondence between the estimated growth rates and reproduction numbers and the forecast, because it's, as I said, a single model that's both doing the RT estimation and the forecasting. Okay, just so to summarize, I think these are four main reasons why generative modeling can be quite useful in this context of wastewater surveillance. And I think, so I've here demonstrated this with respect to estimating growth rates or reproduction numbers. However, I think this similarly would apply to, for example, detecting the presence of a new pathogen in wastewater or just correlating concentrations with some other covariates of interest. Yeah, and if you want to have a look at this model and package that I've been talking about, it's available on GitHub. There's also a package website. And I think, yeah, with that, I just want to thank all the collaborators on this project and looking forward to your questions and discussion.
Thank you, Adrian. That was a very clear, well done presentation. So congratulations on communicating so clearly. Everybody in the audience, please put any questions you have in the Q&A. I have some questions here, so I'm gonna luckily get to be able to ask them. So first of all, I'm really glad that you're doing this work because it needs to be done and there's not enough people with the background to do it. So thank you for all of your hard work on this. I was wondering about sort of, well, the forecasting work that you presented at the end. I think like from a stakeholder or partner's perspective or public health perspective, the most important point of forecasting is when are things going to start to go up again and when are things going to start to go down again. And your video you showed looked really promising that you're able to forecast, but I was wondering if you had a chance to really dig in and see how well the model does at that first uptick, whether it predicts really things are going to start going up, and at the peak, if it's good at predicting when things are going to end. Because that's what when we talk to the media and stuff, they're always asking us to predict the future. So what are your thoughts on the model's performance in those particular spots in time?
Yeah, thank you very much. Yeah, I think it's a really good on-point question. Maybe just let me first say that I think the concentration forecasts I mostly see as an approach to evaluate the model, not maybe something I'm not sure whether it makes sense to report those or share them publicly because I guess the interpretation in terms of public health might be quite difficult. But you're really right that there's a direct connection between how good these short-term forecasts are of the trend when we have this uptick in infections and how good, for example, our estimated reproduction numbers or growth rates are, which would be maybe an early warning or signaling the start of a seasonal wave. And so this is something I have been looking into and also working on summarizing this performance. And I think there it can definitely be said that we have quite a substantial delay in the point when, so retrospectively, the wave actually started and when this can be detected from wastewater. And this delay also depends a lot on the distribution of shedding of individuals over time. So it might also vary between pathogens. And this is mostly because, yes, let's say transmission events happening today only have a delayed signal in the wastewater due to this delayed shedding. So estimating this increase in infections from basically only a partially observed process has some substantial delay always. So the analysis that we've done so far, this can be between 1 or 3 weeks. So it can also then depend on the overall concentration in the wastewater. But yeah, I think you're really highlighting there that maybe when it's actually often easier to predict a trend when you're already in the wave than detecting this first date when you would say, okay, this is when the reproduction number went above 1.
Great. There's a lot of questions in the Q&A and also a question in the chat from one of the hosts. So and I have more questions, but we'll try to get through these. So here's a question. I mean, how do you expect this model to be able to perform as like monitoring frequency goes down, like if it were just once per week? I think you showed some data on this, but can you also tell us the answer to that question? And do you think that this model can just be applied outside Switzerland, or is there something unique about the Swiss data that makes it more appropriate for your model?
So first of all, with respect to the sampling frequency, yeah, this is the kind of analysis that I've shown. And at this point, I'm quite confident that going so I would say that going beyond a frequent sampling of less than 1 week definitely leads to a really loss of signal and like detail of measuring transmission. I think between that, it also depends a lot on the variance of the measurements. And I guess this is maybe then also a bit specific on the laboratory and also other noise factors in the wastewater. So I think this is then maybe quite dependent on the analysis that I've done on the Swiss wastewater. But in our case, we still perform quite well, even with 1 sample per week. Although at the moment, in reality, we are still using 5 samples per week. And I think with respect to generalizability, I think there are some factors which could differ with respect to, say, for example, the sampling method that's being used. Say if they're not composite samples and just grab samples, then the performance could really vary. But the package was really implemented with the idea to allow a lot of configuration. And if you look at the documentation, you can see that there are also we're trying to account for different types of also laboratory analysis that can be done, and also super motivated to extend this in the future.
Great. Okay, we have a question in the Q&A related to that. You mentioned noise. Do you have a sense in your work what proportion of the noise in the data comes from actual measurement noise, or just shedding variation, or even just yeah, like how do you partition the noise? Like in because I know your data was generated, you know, in a laboratory that's been running for a while with specialists, and so your lab methods are really set. But do you have a sense for where the noise is mostly coming from in your data?
Yeah, I guess one important thing to account for there is that it really depends on the concentration that you have in your sample, because that's what I tried to show with respect to the digital PCR. When we are at low concentrations, your noise is really dominated just by the variation in the PCR. So, for example, changing the dilution there can already have a big impact. And something that we don't know super much detail yet is how much additional variation we have basically between biological replicates, so just from the extraction and the preprocessing. But basically, some analysis we did was that basically if you would basically remove all the additional preprocessing or like all the additional noise before the PCR, then the PCR roughly yeah accounts for like 12 percentage points of coefficient of variation. Something I also wrote down in the preprint that I was mentioning. So if you want to look that up. In general, I think there is a much like there's also still a large factor of variation just in the wastewater and in the sampling that we can't address at the moment.
Yeah.
Yeah.
Um, a request from a participant when you're done here to put the link to your preprint in the chat when you have time. And then one more question that I'm going to ask is, have you thought about like your analysis and all of this is for a measurement that sort of is actually quantitative and it's not a rare target in wastewater. Have you thought about like how to apply sort of modeling approaches and some of the ideas that you presented here for targets that are more rare, like Mpox or Ebola or, you know, other very rare high consequence targets like dengue that we are starting to detect in wastewater, not Ebola, by the way, but that was just an extreme example, or avian influenza. Have you thought about how to incorporate like all of this, like noise, et cetera, into interpretation of those types of measurements?
Yeah, this is a really good point. The modeling that I've done so far was really focused on monitoring a pathogen that's already quite present in the population. However, I think that everything related to the modeling of the PCR measurements can be very directly transferred. But the lower part that basically I showed on the modeling, anything related to the infection process, is something that would be probably completely different. So I think the parts that are more related to the actual measurement apply very similarly. Yeah, I think it would be super valuable if people are, also have the time in the future to look into this for example, detecting presence versus absence with sufficient uncertainty measures.
Yeah, it seems like there's enough additional work to do to keep you busy for another decade in
Yeah,
terms
all
of
of us. Yeah.
modeling. Yeah. So, okay, well, I think that should be the end of the questions so we can move on to our next speaker. But thank you so much, Adrian, for that wonderful talk. And I will pass it to Heseth, who will now moderate David's talk.
Thanks, Ellie. Do you want to share your screen, David? Thanks. And you're muted.
Thank you. Can you hear me now?
Perfect. Yeah.
All right. So thanks a lot again for having us here today. Really happy to have the opportunity to speak about our research. Now, my name is David. I'm a PhD student in the computational biology group at ETH Zurich, one of Nico Berenwinkel, who was introduced earlier today. We are working more on the genomic side of things in the wastewater. And today I want to talk about a little bit how we got interested in this, how we got started, and the methods that we have developed to handle this type of data, and what kind of insight we were able to derive on the COVID-19 pandemic based on this wastewater sequencing data. So you remember in December 2020, a few months into the pandemic, we had news from the UK that something there was spreading, a new genomic variant, and it was spreading fast, right? So we were trying to find it here in Switzerland in our sequencing consortium that we had in the country. Our job in the group I'm in was more on the bioinformatics. We were getting these sequencing results, analyzing them, computing consensus genomes from it and aggregating the results. And at that moment, during this alpha introduction period, we thought, okay, why not try and turn to these wastewater samples that our colleagues are working with? Maybe we can sequence something from there and get some insights. So we did a pilot study in some of the main cities of the country, but also touristic regions in which the variant might have been introduced earlier. We applied the same sort of full genome sequencing with the tiling amplicon protocol, the Arctic protocol, which was already used on the clinical genomes. We had to, of course, adapt it somewhat, but most of all for us, we had to come up with new computational methods that made us able to detect it earlier, up to 2 weeks lead time in the main cities that we looked at. Right, so now, how did we do this? And how does it differ from the way we analyzed previously clinical samples? Well, generally in a clinical sample, you sequence it, you get an alignment of the reads on a reference, which is what you see on the top part. And you want to know which strains of the candidate strains that you have on the bottom part here you might have in your alignment. What you do is you call mutations on the different reads, and then you construct a consensus sequence out of it, which is just the majority nucleotide at every position. And now this consensus sequence, you can compare it with a database and say, this is probably that variant that I have in my sample. Now, this approach assumes that you have one dominant strain in your sample. It cannot work for a mixed sample or high mixture wastewater sample, because you might construct an artificial sequence that actually doesn't exist. Right? So if you go back to the alignment now, you say, okay, maybe I cannot have the full haplotypes of what is in my sample, but I can go and look at reads or read pairs where I have some local haplotypes. So if I have more than one mutation on this read pair, if I have a string of co-occurring mutations, I can have a bit more confidence in the fact that I have detected a certain variant which has this string of mutations in its definition. Right? So we built some computational method to do this, which is called Kojak. It indeed enabled us to detect the alpha variant early, but also the following variants. So this is an example with the delta variant, which we also were able to detect earlier in the wastewater samples in general in many different cities. And also we had the first detection of Omicron in Switzerland, and others used it. Here you see, for example, to track the spread of Omicron in the English wastewater, and you can quite clearly see it spreading from the capital to the main urban areas, and from there to the rest of the country. Now, we were quickly convinced that sequencing these wastewater samples was indeed informative, but we wanted to know how much information we could extract from this. Can we go a bit beyond? Can we be quantitative in this assessment? Can we reconstruct the relative abundance of the different variants? So to try and tackle this problem, let's go back to the alignment that we've seen earlier. You cannot directly observe the relative abundance of these different variants, but what you can measure is at each different position, what is the fraction of different mutations. And now these different mutations, they are related to the relative abundance of the different variants, but they are related in a way that you cannot aggregate these results so straightforward. You cannot just average over the frequencies of these mutations, because a given mutation might be characteristic of more than just one variant. So you have to take that into account, and you can take that into account in a statistical model, which makes the assumption that the relative frequency of a given mutation is going to be expected to be the sum of the relative abundance of the different variants that have this mutation. Right? So that's quite a straightforward assumption from which you can derive some sort of linear probability model, which you can make statistical inference on to try and get the relative abundance of the different variants. Now this works really well. It works really well when you have good data, and usually we get good data from the people we collaborate with, but you know that wastewater sequencing doesn't always look like this. Often it much more looks like this. You have some regions that are not covered in the genome because you're sequencing from a challenging sample. When that happens, you cannot get the mutation frequencies everywhere. And now, if that happens and you have these dropouts in too many regions of the genome, you might not be able to solve uniquely this statistical model, and you cannot identify the relative abundance of the different variants anymore. So this is quite visible in this simulation of relative abundance of different closely related variants. As you see, when there is no missing values, you can deconvolve and get the baseline values. But as you increase the missing rate, there is a tipping point after which the problem is not solvable anymore, and you just lose the signal completely. So these missing rates at which this breakpoint happens might seem high, but those are things that we can observe, right? Especially when we go at low incidence, we get low concentrations. And at low concentrations, we have a lot of noise, as Adrian explained earlier. And when we got this high noise, we get high dropout values. Now, we still want to be able to quantify the relative abundance of the different variants, even when we have these high dropout levels. So how do we tackle this? Well, we make use of the whole time series structure of the data. So we don't consider a single sample at a time. We concatenate the whole data together, and we add one simple additional assumption. We say, okay, we assume the signal must be somewhat smooth and in the variant relative abundances. So the variant relative abundances, they should evolve somewhat continuously, which can be enforced by adding some penalization to the statistical model. And this small penalization actually makes us able to deconvolve even when the missing values are extremely high. And so we can recover the relative abundances even with very sparse data. Now this is in a computer package we call Lollipop. It's integrated into our viral NGS analysis pipeline VPipe, and we've been using it for the national variant monitoring program, which has been running from early 2021 up until now. And we are producing these reports on the relative abundance of the different variants, and we are reporting to health authorities, hospitals, and the public in general. And they're really interested in this data, but why are they interested in this data at all? If we take a step back, why are we looking into these different variants? And why is that relevant for public health at all? Because most of the variants, they are inconsequential and they don't really change the course of the pandemic. Like many different variants have emerged, but sometimes, and we've hinted at this in the beginning, sometimes there is a variant which can have an increase in transmissibility or can escape immunity conferred by previous vaccination or infection. When that happens, this variant can have some advantage in its fitness. And if you look at here on the right for the alpha variant, if you don't look at the data stratified per variant, you just look at total incidence or even total loads, things might look like they are okay, the situation is stable or even improving, getting better. But actually, if you look at the data stratified per variant, you very quickly can see that something wrong is happening. And you might have some sort of epidemic within the pandemic, which is brewing. And you might very well be facing a large number of cases and hospitalizations in the short-term future. So it's very important to try and estimate this fitness advantage of a new variant as early as possible. And people have been working on it extensively during this pandemic on the clinical data. Generally, you assume some sort of survival of the fittest type of evolutionary model, which says a variant has a fitness. If its fitness is greater than the average population of variant at a given time, is going to replace them. And if it's much more fit than the other variants, it's going to replace them rapidly, right? So this sort of dynamics can be seen here, simulated from this model on the right. And this type of model, you can fit it so on the clinical data, but we've shown that you can also fit it on the wastewater relative abundances of the different variants. So this is what you see here, a fitted model on the data from the wastewater in Switzerland. And the parameter estimates of such a model are the the fitness advantage between different pairs of variants which have been observed in competition together in our samples. So you can see, for example, on the top left, that Omicron BA1 had a tremendous fitness advantage over Delta, which it replaced. And you remember that the massive waves of infections that Omicron caused when it spread in the population. And then you can assess some sort of the epidemic potential, in some sense, of new variants as they come through. Right. So those are estimates that are important for our public health partners. We can also obtain them from variant-specific digital PCR assays. Our collaborators at EAVAG, they've developed some of these TEM probe assays, and we have developed computational methods and R packages to analyze them and compute estimates of the fitness. They usually correlate really well with what we can obtain from clinical data when we have clinical data, because it's much more efficient to compute this from wastewater samples. With just a handful of wastewater samples, you get the same information content in general as from thousands of clinical sequences, for which you need at least tens of thousands of positive tests, which we don't have anymore, right? I don't think anybody is testing that extensively anymore. So we can rely on estimates based on the wastewater data. From this sort of model, we can also compute short-term forecasts of how we think the situation might evolve in the near future in terms of the composition of variants in the population. And that's also something that can be useful. Now, to finish, I just want to speak also a little bit about shedding. And I know this is a subject which is important in the community. So shedding, just to briefly go through this, describes, or the shedding profile describes how much of a pathogen is shed throughout the infection time of a typical, let's say, infection, right? And now this is important because the quantities we are interested in, such as, for example, incidence, we cannot observe them directly from the wastewater samples, but at best what we can observe is a convolution between the shedding profiles and those quantities of interest, such as incidence. Now, this convolution is going to have an impact on the signal. It's going to scale it, first of all, it's going to shift it a little bit, and it's going to smooth it a little bit too. So in general, you need to calibrate some models or explicitly take that into account for these models. But what can be a further complication is that different variants can have, and have been observed to have, different rates of shedding. So they can have usually different total shedding, but you could also imagine that they could have different shedding dynamics. So those are going to result in additional biases, especially if you have calibrated a model assuming a certain type of shedding, but now a new variant is coming with a different type of shedding. This might add a bias to your estimates of incidence that you're trying to obtain. Now, with the genomic data, we're also interested in this, for example, fitness estimates. And I just want to say first that this is not just theoretical. We've observed sometimes this decoupling in the new cases from wastewater loads. For example, during the epidemic of Omicron, and Omicron was apparently shed less in feces than Delta, which meant at the time that the loads we observed were some sort of an underestimation of the new cases based on what was previously calibrated. So that's problematic, but we could show that for the fitness estimates, this didn't matter in fact, and that the different shedding profiles result always in the same fitness estimates. This is based on the common assumptions of the evolutionary models that we are using. But indeed, from the data that we had, we were still able to estimate early the fitness advantage of Omicron. And when we had then the clinical data, we could confirm that those estimates didn't show any bias in the different locations that we surveyed. Right, so to wrap up a little bit what we concluded from these little experiments around shedding is say, if you are interested in metrics on some sort of absolute scale, then shedding and normalization is something that is very important, and you should think about it in your model a lot. Then if you're interested in metrics on some sort of differential scale like fitness advantage, then it's not really important. And at least for fitness, it actually doesn't have any impact. And for growth rates or reproduction numbers, for example, models of reproduction numbers such as Adrian's model or Jana's model, they are very much robust to changes in shedding. So even if they are misspecified in that sense, it will incur a bias which is much more benign than what can be what can happen on the absolute quantities, right? And it will tend also to recalibrate itself after the wave. All right, so to wrap up, we are working on extending analysis and modeling pipelines that we have also to new protocols for new pathogens. And we are working on different ways we can do things together that will make our methods more accessible to a larger number of people who want to do this. And with this, I want to thank all of the fantastic people we've been working with in this collaboration. It's a real pleasure to work with these talented people. And that's it for me. If you have any questions now, I will be very happy to take them.
Thank you, David. That was also a very clear presentation. So thanks for that. So there are two related questions here, so I'm going to try and kind of combine them. Basically, in your fitness models, I guess, is it possible to use information about the mutations to predict like the combinations of mutations or specific mutations that would result in large fitness gains?
Thank you. That's an excellent question. That's actually, some sort of $1 million question, right? Many people have been looking into this. Can you predict the phenotype of a new variant when you have its genotype? And if I'm up to date on the news, the answer still seems to be no. It still seems like we cannot really predict what will be, the effect of a group of mutations. I mean, some of them that we've seen coming in multiple variants independently, and there is convergence, and we know a bit what sort of effect they can have, but in general, this is, I don't think modeling for this seems to be advanced enough.
So the for the forecasting portion of the talk, are you using any of the information about the variant, such as fitness or changes in proteins or things like that?
So here it's just from the fitness that has been estimated from the wastewater data. So it's just derived from the time series of relative abundances. And we can, if I go back to this slide, we can use the observed portion of the time series up until some point to fit those curves. And from this curve fitting, you can estimate the fitness parameters of those, the relative fitness parameters of these different variants. And then you can forecast in the near future, right? But of course, when you do this, you assume that things are going to stay the same, at least, for example, in the number of variants. You cannot predict that a new variant that you don't know about it yet is going to come and maybe sweep everything. So this is only some short-term type of forecast, right?
Yeah. So the forecasting is agnostic to information about what the mutations mean, I guess.
Yeah,
is kind of
absolutely. You don't, for this, you don't need it.
Yeah. Okay. So I think a lot of people spend a lot of time thinking about shedding. And so in some ways, it's a little bit sorry, surprising that the shedding would not affect the transmission dynamics that are inferred from the wastewater. And actually this to some extent relates to some questions that came in the chat to Adrian. I guess when you were evaluating this, did you kind of bind the total effect of, or the total changes that were possible in shedding dynamics in order to evaluate whether or not they could impact things?
So this is of course based on some assumptions of the evolutionary models that we use, right? But in general, we sort of let's say if you take, for example, a halving of the total shedding, right? It's going to have a huge impact on the absolute loads that you observe, right? It's going to, on average, halve them. But if you're observing something that doubles, every week, if you'd only observe half of it, it's still going to double every week, right? So some metrics on a differential scale, like growth rate or something, are not affected by this type of biases. Now, if you consider arbitrary changes in shedding, not only scaling of the shedding profile, but different dynamics. It gets a bit more complicated. It depends on the assumptions of the selection models that you have. And we also are looking at what happens if you drop some of these assumptions. So usually when you drop these assumptions of these selection models, of course your selection estimates are going to be wrong, but they're going to be wrong whatever you use to estimate them, right? And we want to see if they are more wrong if you look at wastewater with the wrong shedding model than they would have been if you had access to the actual relative incidence of a given set of variants. And so in general, we observe that when we look at this, and you can look at it, you can derive some of these properties. Indeed, it's robust to these changes in shedding.
I'm curious if your program has applied these models to other pathogens like influenza, or like any other pathogens, and looking at variants of different things.
Yeah. So we are not routinely sequencing influenza or other pathogens at the moment. As Adrian has shown, we are looking at quantifying their loads with digital PCR. I mean, our colleagues are doing this, and Adrian is modeling it. But now we're not doing NGS in the routine monitoring program at the moment.
Can you comment on the differences between Lollipop and other deconvolution methods like Phrasia?
Like Freya, yeah, it's so the approach is very similar. So there is now like a whole zoo of different methods, and it's really great to see that how it went from, oh wow, we have this sequencing reads. What do we do with them? We have to come up with something to fast forward a couple of years, and many people have looked into this, and many interesting solutions have come up. We have participated to some benchmarks, like single-blinded benchmarks, or we've tried a few ones. In general, if you have a good sample, many of them work and will give comparable results, and I think that's good. That's reassuring. That it seems like it's a problem that many people are able to solve. Then, depending on the specific issues that there might be with the sample, depending on the specific noise, some of them might be robust to more types of noise. So I would advise anyone to try different one of these methods. Some might be more accurate for the quantification of relative abundances, but miss the low-level variants. Some of them are better for another. I mean, there's still, I believe, some research that is needed in this area.
On the benchmarking question, have you had the opportunity to work on any data outside of Switzerland? I guess this question was also directed towards Adrian, whether or not the model also, how robust both of these models and forecasts are to other types of localities, which might have less frequent data.
Yeah, that's an excellent question. We've tried some of our methods on data from other countries, and I guess that's always valuable to see with the different different things that can happen. And also for our own sample, I mean, we were sequencing, we're daily samples at first for some time, and then we ran some experiments to see, can we downsample? This is, taxpayers' money. Can we get something that is almost as good for significantly cheaper? And so, we're looking at this at how robust is are those estimates to different sampling schemes, fewer locations, fewer time points. In general, I think for quantifying the dynamics like this, if you have a measurement that is, precise, you don't need as much time steps. If you have a measurement which is noisy, you need more time steps. I think that's not surprising, but beyond that, maybe it depends on more of the specifics. But
on that note, I guess you've said a couple times, like both in the presentation and in the Q&A, about like, oh, if you have good data, everything is beautiful, right?
Yeah.
Everything works. But I guess I'm curious, like in your experience, like what protocols or things, sequencing approaches are more likely to give you good data that are easily analyzed for these variants and have good coverage across the SARS-CoV genome? Or is it, a messy sample and there's inevitability in that some samples are just not going to provide you good information?
So it definitely depends on where the sample comes from, in some sense. That this I cannot really comment too much on, but it's evident that some of them have more inhibition. Then your PCR amplification is not going to work so well. You're going to have more dropouts, and then you can still obtain these estimates if you have a model which is robust, but you're going to have less precision. Now, my colleague Annika has looked into pretty much all of the sequencing technologies and compared them with our wastewater samples and also spike-in experiments. And in general, what came out of this is that you can use reliably, at least on our samples and our spiking experiments, like all of these sequencing technologies. The cheaper ones, of course, will have more noise. But if that's something that is not too prohibitive compared to the reduction in cost, then we would say go for it. Yeah.
Awesome. Well, thank you both for this amazing presentations, and it was great to learn more about some of these modeling approaches. So I appreciate your time, and we'll let everybody get on with their day. And I think Ali already shared the other preprint in the chat in case anyone needs that. And then I think we mostly covered things. I appreciate, Adrian, you answering this last question in the chat. So thank you all for your time.
Thanks for having us.
Thanks a lot.